Tuesday's quiz will cover one lecture, tonight's lecture, which means since tonight's lecture is about genes and mutations, it means you really have to have a pretty good handle on transcription, translation. In other words, in order to be able to figure out what kind of a mutation you're dealing with, and you'll see what I'm talking about as we're going through this material tonight. So tonight's lecture will be the content for the quiz on Tuesday. Relative to the laboratory, how many of you have potential access to a Mac? Right. So if you don't, maybe you know somebody who might. What I'm thinking about doing is putting up on the website a step-by-step how to do the exercise that we were supposed to do in lab on Tuesday night, which is basically access AB1 files, which are the electropherograms for physiological unknown 2, and then walk you through the process for determining what organism type you have for physiological unknown 2, because that's how we're identifying them. We're using 16S ribosomal RNA nucleotide sequence. You're not doing any biochemical testing. You're not doing gram stains. In fact, you didn't do anything with those organisms other than take a little bit of DNA out and put it in food, and then for some of you beat the heck out of it with lab feed. So anyway, we'll see how that goes, and if people just have too much trouble with it, we'll still have some time in class to try to access those sequences. Anyway, they're on the website. They are available. I have had students just use the instructions that are in the lab manual and access the information and do that assignment at home, so I know it's doable. But you have to have 4 peaks. If you have a Mac, you have to have 4 peaks, which is free. It's a software that allows you to open AB1 files so that you can see electropherograms, and you also have to have a program called Stuff It Expander, because those files are shrunken so that they fit nicely on the website. If you're using a PC, I don't know what the equivalent software is. I don't know. I think the 4 peaks equivalent might be called BioEdit. And actually, maybe Stuff It Expander is the same regardless of what kind of computer you have. You have to be able to expand those files. They're not zip files, they're just stuffed. Stuffed files. Anyway, so I'll put some information up on the website, and hopefully that'll. If that doesn't work, we have a couple of labs coming up where we would not normally be here the whole time, so we can kind of work that in. Yeah, might work that way. Okay, so last time I had this slide up, and everybody looked like you were just completely exhausted because we had a lab exam, and it was fairly evident nobody wanted to hear about CRISPR again. But then I thought, well, this is important. And so here it is again, CRISPR. So a little more information about the CRISPR-Cas system. So this is actually recognized now as a type of adaptive or acquired immune system for prokaryotic cells. It's been found in most bacteria that have been identified. It's also been found in archaea. So it's allowing these organisms to recognize some foreign agent that's going to get into them after it has gotten in one time. What they do is basically steal DNA segments from the invading organism and incorporate that into their genome. So CRISPR, the letters stand for clustered, regularly interspaced, short palindromic repeats. Who makes these names up? It turns out palindromic sequences, we're going to talk about a little bit later because we're going to talk about restriction enzymes. And the restriction enzymes, inverted repeats, really are palindromes. A palindrome is a word that reads the same forward and back, like pop, radar, rats live on no evil star, a manaplan animal, something like that. Anyway, yeah. Turns out that the ones that are associated with the CRISPR system aren't actually palindromes. They're just nucleotide sequences that kind of have some recognizable repeat patterns. And so somebody recognized that these things were there, and then between them, there were these odd pieces of DNA. They couldn't quite figure out what they were. It turned out there's what they called spacer, these pieces of DNA that were between the interspaced or regularly interspersed palindromic repeats. These spacers were actually DNA that had been taken from an invading virus. So what virus is doing, we haven't talked about viruses yet, But viruses that infect bacteria typically get into the bacterial cell, and then they take over the cell's function. Bacteria have different mechanisms to defend themselves. They use restriction enzymes, and it turns out that actually the CRISPR system is a type of restriction enzyme, so that if they can recognize viral DNA as foreign, and they can chop it up before the virus has time to take over their function, then they can get rid of the virus and they can survive. So this is what is happening. The cells are actually taking part of the viral DNA, incorporating it into their genome as these little spacer regions between the palindromic repeats. There are quite a number of proteins associated with this system. They're called Cas. Cas stands for CRISPR-associated. CRISPR, capital C-A-S, is associated. These are proteins that are encoded by genes somewhere near this set of spacers and repeats. Turns out there's a whole bunch of different Cas proteins. If you're paying attention to what's going on in the news, most of the emphasis is on Cas9, because Cas9 is being recognized as something that can be used for gene therapy, but there's a whole bunch of these things. They have different functions. So here's this little, this actually came off the internet, you can look this up. There's all these different proteins, so there's Cas1 through 10, and they have different functions in association with this CRISPR system. What we're mostly interested in is these Cas proteins that are guided to foreign DNA by what are called CRRNA molecules. So the CRRNA is CRISPR RNA. We talked about transfer RNA, messenger RNA, ribosomal RNA, and then in prokaryotic cells we've got this CRRNA. The CRRNA is CRISPR RNA. And these CRISPR RNA molecules are encoded by viral genes. So the bacterial cell or the archaeal cell will take some viral DNA, incorporate it into their genome, and it stays there. Not necessarily permanently, because what they're finding out, especially in archaea, there's gradual turnover. This DNA, they incorporate it, and after a period of time they may lose sections of it, but the incorporated DNA encodes the CRISPR RNA. And then the function of the CRISPR RNA is to guide these proteins, these Cas proteins, that are endonucleases. Endonucleases are enzymes. Again, notice the ASE ending on it, so you know it's an enzyme. But an endonuclease is capable of breaking phosphodiester bonds. So they will cut DNA. Sometimes they'll cut one strand, sometimes they cut both strands. This is shown with a little pair of scissors. If the CRISPR RNA guide these Cas proteins to a viral genome that has just gotten into the cell, then what the cell can do is chop up that viral DNA before the virus has a chance to take over. And so that's what they're doing. They're using these little short CRISPR RNAs to guide the protein to a specific site. And because the RNA was encoded by the viral gene in the first place, that RNA is going to be complementary to a specific region on the virus. It's going to guide the protein to that site, and the protein's going to cut. So these Cas proteins can destroy the viruses that get in. And again, this business of them recognizing it when the virus gets in a second or subsequent time, this means that the bacteria has to have had some experience with the virus in the first place and have survived. But that's kind of how our immune system works also. If you encounter some kind of a foreign agent, your adaptive or acquired immune system will allow you to be immune the second or subsequent time you run into that same agent. So it's kind of a fascinating system. The Cas9 is the one that's particularly of interest because humans recognize that we can engineer RNA molecules, and as a consequence, we can make RNA molecules that will guide the Cas9 proteins to any site that we want. So basically what we can do is take the CRISPR RNA, synthetic CRISPR RNA, if you will, and it will guide those Cas9 proteins to a specific site. This is talking about cutting out what are called indel mutations. Indel stands for insertion or deletion. Something is damaged, some piece of the DNA is damaged, there's a potential for taking the Cas9 protein to that specific site and cutting out that damaged DNA, so that it has tremendous therapeutic potential. So not too surprisingly, when people find out about something, then they start playing about with it. So there's a lot of different variations of Cas9. Some cut both strands of the DNA, some only cut one strand. A lot of times if there can be a mutation recognized and it's only on one strand, if that piece of DNA is cut out, the cell will automatically replace it with the right DNA using the complementary strand. And then they can do other things with it, guide the Cas9 protein to a specific site, and then maybe the Cas proteins can recruit other proteins that can come in there and do something specific. So our main point here is to pay attention to the CRISPR RNA, the cRRNA, because the cRRNA, that's RNA that is actually encoded by viral DNA, little segments of viral DNA, and these cRRNA's function is to guide these proteins, these endonucleases, to the viral DNA and chop it up. Okay, enough of that. So our actual topic for tonight is genes and mutations. And again, since this is the only lecture we're having this week, next week's quiz is on this lecture. Which tells you that CRISPR stuff might show up earlier than that, because you've seen that before. Okay, so what is a gene? I'm sure you've heard the term, right? Anybody?
A segment of DNA.
Right. So a gene is a segment of DNA. You're pretty safe if you say that the gene is a segment of DNA. And you can see in that illustration, there's chromosome there. Then you see the DNA kind of winding out the top, and then it magically becomes a single strand of DNA, which obviously wouldn't, because we know that actually the chromatin is DNA wrapped on histone proteins, and there's other heterogeneous proteins associated, the enzymes and so forth. But anyway, a gene is a segment of DNA. We have to get more specific when we start incorporating viruses into our thought processes, because some viruses, the genome of some viruses is just RNA. Consequently, a gene can be DNA, but it might be RNA. So you have to remember that we have two different types of nucleic acid. Now, DNA is the primary information storage material within cells, but in some viruses, all they have is RNA, and it's still considered a genome. We haven't got that term yet. So, more specifically, what is a gene? Another way we could define the term gene is to say a gene is a unit of heredity, which is kind of interesting because at the time that the term was developed and people were talking about heredity, they actually had no knowledge of DNA. So they didn't know that nucleic acids were the hereditary material within cells. So this term gene was actually coined long before people had any idea what a gene really was. The idea of inheritable units was proposed by Gregor Mendel. If you've taken any kind of general biology class, you've undoubtedly heard about Mendelian genetics. So Gregor Mendel developed these, actually he did a lot of work with pea plants and crossing pea plants and looking at characteristics of pea plants and said there was some kind of hereditary unit. He didn't actually use the term gene. The term gene kind of developed over time. So Charles Darwin developed a hypothesis for inheritance that he called pangenesis. This was in 1868. And the term genetics was first used in 1905. Genetics is the study of heredity and factors that are passed from one generation to the next. Again, at the time, nobody really knew what that material was. But investigators used the term pangene from Darwin's work, and then that was later shortened to gene. So the term gene was actually used first as that term in 1909, this unit of heredity. But at that time, people didn't know what it was, because DNA wasn't discovered until 1952. Or I should say, the structure of DNA, the composition of it, wasn't elucidated. Is that the right word? Well, anyway, until 1952. Which is not that long ago. Before that, people thought that the genetic material, the hereditary material within cells had to be protein. And the reason they thought that, because proteins are much more complex. You have a lot more different types of amino acids than you have nucleotides. Anyway, so all this tells us is that the composition of a gene and what a gene is, this has changed a lot over time. Again, the idea that it can be DNA, it can be RNA. You're talking about some viruses, it is RNA. So that's something. The other thing that happened, there was a period of time where all genes were defined based on structural genes. Now we call them structural genes if they encode messenger RNA, and then that messenger RNA is translated to yield a polypeptide. But not all genes do that. So structural genes do. And there's a relationship between the term structural gene and the term cistron. So remember we talked about polycistronic versus monocistronic? So a structural gene, again, that's a section of DNA that's going to encode messenger RNA. That process is transcription, and then the messenger RNA is going to be used to build an amino acid sequence. There was quite a period of time where that was the definition of a gene, and there was this one gene, one polypeptide. That was the story. All genes encoded polypeptides. But the definition of a gene has changed over time because they don't anymore. And if we were to ask this question, do all sections of DNA encode messenger RNA? What do you think? Is messenger RNA the only kind of RNA? No, because there's ribosomal RNA, and there's 3 different flavors, if you will. If you talk about prokaryotic cells, you've got the 16S, the 23S, the 5S. If you're talking eukaryotic cells, the Svedberg units, those values are slightly different, but there's 3 different sizes of ribosomal RNA. Then there's transfer RNAs, and there have to be a bunch of different transfer RNAs because they have to have different anti-codon regions that will allow them to line up and hydrogen bond with the right codon so that the amino acids get put in the right place. There's the small RNA. Some references refer to those as small nuclear RNA, the ones that make up the spliceosomes, because spliceosomes are in the nucleus. And now there's CRISPR RNA, so there's a bunch of different types of RNA. Anyway, so no, not all sections of DNA code for messenger RNA. There's all these other types. Well, for a while, those weren't considered genes, but they are now. So there are specific genes that encode ribosomal RNA and transfer RNA, et cetera, et cetera. So we could just say a gene is a segment of DNA that encodes RNA, but that leaves out our regulatory units when we're talking about operons, because in the operon you've got a promoter region and you've got an operator region, and sometimes there's another section in there that we didn't talk about, attenuator. So what are those things? Those are sections of DNA. They don't encode anything. They're not transcribed. So if the DNA encodes messenger RNA, it's going to be transcribed, and then the messenger RNA is going to be translated. If the DNA encodes ribosomal RNA, transfer RNA, CRISPR RNA, those things are transcribed, but they're not translated. Once you get to RNA, that is the product. And sometimes that product can be a catalyst, because remember the 23S is the ribozyme peptidyltransferase, and the small nuclear RNAs, those are the ribozymes associated with post-transcriptional modification, taking out the introns, hooking together the exons. So they have a function, they're just not going to encode protein. Those things, the promoter and the operator, I'm not sure if those are considered genes or not. But maybe they are. I don't know. There's regions on DNA that are called intergenic regions. So there's regions between genes. I'm not sure about those guys. Anyway. Yeah, what about those? I don't know. So we'll just go back to this definition. A gene is a segment of nucleic acid. And we'll go on to say a gene is a segment of nucleic acid that has a specific function. It can be DNA. In cells, it is DNA. But in some viruses, it can be RNA. And sometimes they're transcribed and translated, and sometimes they're not. So then there's some other terminology that goes along with this section. Once we get a gene defined, this segment of nucleic acid that has a specific function. If you have alternate forms of genes, these are called alleles. When we think about alleles in association with humans, we often think of things like eye color, because people are familiar with eyes. They come in different colors: brown eyes, blue eyes, green eyes. Put contact lenses in them, you can make them yellow and violet and all kinds of different colors. That's not genetics. But anyway, alleles are alternate forms of the gene. This means the DNA is homologous. Homologous DNA is DNA that has basically the same function, but the sections are not identical. So the nucleotide sequences are going to vary just a little bit. In this particular example, you see these things that kind of look like cigars with dents in the middle. Those are supposed to be chromosomes. And at a specific location on this chromosome, we have a gene. So this gene is here. This particular allele encodes white flower. This particular allele encodes purple flower. They're in the same position. The term locus is used to determine or to describe the position of a gene on the chromosome. They have a specific locus, that's where they're located. But these would be DNA encoding the same general aspects, that is flower color, but they're not identical. Another example would be bacterial flagella. We know that flagella are made out of flagellin proteins. Flagellin proteins don't all have the same amino acid sequences. So you can have slightly different flagellin proteins that are encoded by alleles, homologous DNA, with slightly different nucleotide sequences. Again, the term locus, that's the location of a gene on a chromosome or within an operon. This suggests that genes stay put. They kind of do. There's quite a lot of information about gene mapping. You can look up gene maps, which show you the locations of genes on chromosomes. They tend to stay put. On the other hand, they can't move. And we'll see about that in just a little bit. So the locus is the location of the gene. And then genetics. So all of these are the kinds of things you can expect to see on the quiz, either definition or fill in the blank. There's a lot of material here. So genetics, again, this is the science or study of heredity and the factors that influence variation observed within organisms. These factors are passed from one generation to the next when we're talking about heredity. As I said, for a long time, people didn't know what those factors were. Now we know they're segments of nucleic acid. So classical genetics, again, if you study general biology, you probably spend some amount of time studying classical genetics or Mendelian genetics. And this is based on the work of Gregor Mendel. You get to make Punnett squares and you get to do genetic crosses and see how this all works out. Well, Gregor Mendel was doing these studies with pea plants back in the 1800s, around 1860. And again, at that time, he talked about units of heredity, but he didn't really use the term gene and didn't really know what genes were. He did talk about some characteristics being dominant and other characteristics being recessive. So if a characteristic is dominant, what that means is it will mask a trait that is recessive. I can tell you right now, if you look at my eyes, they're brown, but I have a son with blue eyes. Blue is recessive, brown is dominant. I have to have both genes, which means I'm heterozygous. I have two different colored alleles, one blue, one brown. Otherwise, I couldn't have a blue-eyed child. Although it's not quite that straightforward, but anyway. So there are dominant traits. Brown is dominant, blue is recessive. If an organism that is a diploid has two alleles that are the same, so if I had two brown, then I would say that I was homozygous for that particular characteristic. Remember that homo means the same, and what this refers to is what kind of zygotes can be produced. Actually, it has to do with what kind of gametes can be produced, and those are going to combine with other gametes to form a zygote. But homozygous means that the alleles are the same. If an individual is heterozygous for a particular trait, it means their alleles are different. And there's lots of variations on this theme. Later on, we're going to be talking about blood typing in the lab, and kind of a common way to describe the ABO blood groups. You have A-type blood, B-type blood, AB-type blood, or O-type blood. If a person has O-type blood, they have two alleles, and those alleles are the same because O is recessive. If O is in combination with either of the other alleles, the A or the B will be expressed. So if you have the A-O combination, your blood type is A. If you have the B-O combination, your blood type is B. If you have A-B combination, your blood type is AB. The only time you can have O is if you're homozygous for the two recessive alleles. Of course, now, the new textbooks, they've stuck another letter in there that causes all kinds of confusion. We'll get to that later. So, Punnett squares, these things were developed by R.C. Punnett, and they're used a lot to describe Mendelian genetics and what's going on. How many of you have played with these things? Probably most people. You probably all had some general biology class at some point in time, and so you get to see these crosses, you do genetic crosses, and there's ratios of how many offspring you're going to get of a certain type. And never mind, because we're talking about bacteria and they don't do that. So now, when you study genetics, most often, you're not really interested in Mendelian genetics so much. What we're talking about is genes, and we're looking at genes at the molecular level and trying to understand how heredity is really controlled at the molecular level. And you may have gotten that feeling because we keep going back to DNA and RNA and nucleotide sequences. There's a tremendous amount of information being accumulated here. So again, my job is to try to sort this out and present that which I think is appropriate for an introductory class in microbiology, which gets tricky over time. There's another term that you need to be familiar with, the term genome, but different references will give you slightly different versions of what is genome, because some references will tell you that the genome is the total DNA content of an organism. I'd agree with that, except that in an organism that is eukaryotic, if that eukaryotic organism has mitochondria or chloroplast, the mitochondrial and chloroplast DNA doesn't belong to that eukaryotic organism. The mitochondrial DNA, the chloroplast DNA, that belongs to a prokaryote that was incorporated into the eukaryotic organism at some point. Consequently, I'd be willing to say genome is total DNA content of the chromosomes of an organism, not total DNA content. Because you can't count mitochondrial DNA. My mitochondria, their DNA is theirs, and it's prokaryotic. Even though they've passed a lot of that to me. By that I mean eukaryotic cells. The eukaryotic cells are now holding most of the DNA from the mitochondrial genome. They're holding most of that in the chromosomes of the eukaryotic cell. Well, anyway, genome is still, we're going to say genome is total DNA content of the chromosomes of the organism. So mitochondrial DNA doesn't count, chloroplast DNA doesn't count, and plasmid DNA doesn't count because plasmids are extra chromosomal. Gets a little tricky when you're not sure whether you're dealing with an organism that has only one chromosome or if they have multiple. But for our purposes, DNA of chloroplast, mitochondria, and plasmids is not genomic. Genomic DNA is it's the total DNA of the chromosomes of the organism. These terms you're probably familiar with, genotype and phenotype. But oftentimes, you'll think about them a little bit differently if you're thinking prokaryotic organisms versus eukaryotic organisms. So genotype, that's the genetically determined characteristics of an organism or the genetic potential. What did we find out about last time that you get to have a quiz on this time, where organisms can have genes, but they're not being expressed? In the operons, you can have operons that are off, they're repressed, which means the genotype is not necessarily expressed all the time, because you've got a potential, you've got a genetic potential, but those genes may or may not be expressed. The same is true with humans. If you think about our cells, if the original cell, the fertilized egg, that grew into a multicellular organism, there's a lot of DNA that's not being expressed in many of our cells because you have blood cells that have particular characteristics, muscle cells that have particular characteristics. They're different. In theory, they all have the same DNA content. Technically, they don't, but if they did, it's not all expressed. Some of it is, some of it isn't. So genotype is genetic potential. May or may not be expressed. Again, why? Well, because sometimes genes are repressed. In eukaryotic organisms, where you have multicellular organisms, exactly what is controlling that? Remember those small interfering RNA molecules that are supposed to be having some regulatory function? So we don't really know exactly how things are controlled. Another term is phenotype. Phenotype is the observed characteristics of an organism or a population of organisms. Again, when you think phenotype of a eukaryotic organism, a multicellular organism such as humans, you often think phenotype would be eye color, hair color, blood type. It's the genotype expressed. But when you're dealing with prokaryotic organisms, you're not going to see those kinds of characteristics from a single organism. You're going to have a whole population of organisms. You're kind of used to working with bacteria where you have a lot of them. You're dealing with more than one. But phenotype is observed characteristics. It's the genotype expressed, but what kind of factors can influence phenotype? Whether or not things get expressed. Well, you can think about operons. So you can think about inducers. You can think about repressors. Are the repressors active alone? Are the repressors going to require a co-repressor? So there's that molecular level stuff going on. But the other thing that we found out, because we did an exercise when we were working with E. coli on differential media, you recall Escherichia coli is the organisms in your colonies that are on the left-hand side. So Escherichia coli on T7, the colonies are yellow. Escherichia coli on MacConkey's, the colonies are bright pink. Escherichia coli on EMB, the colonies are dark with an iridescent green. So what's another factor that's going to influence phenotype? The environment. Exactly. The environment. And we know that environment can be inside, as I said, repressors, inducers, that kind of thing. But the environment can also be outside, external factors that can influence whether those genes are expressed or not. Needless to say, there's a lot between organisms being exposed to a particular chemical and whether genes are turned on or not. We're not going to get we can't get into all of those details. We did talk about quorum sensing, where specific chemical signals are picked up by cells, and those chemical signals can be used to turn on genes, that is, cause genes to be expressed. There's a lot of factors going on. There's a lot of things happening. So this is kind of just brushing the surface. Environment is a major factor. Now, the other thing is that the genome, total DNA content of the chromosomes, and consequently the genotype, this is not necessarily static. Well, we know it isn't static because we talked about characteristics of life, and one of the characteristics of life was mutation. Mutation is any change in the nucleotide sequence of the DNA content of an organism. If you're talking about viruses, which technically aren't organisms because they're not cellular, a mutation can be any change in the nucleotide sequence of RNA. When we're dealing with cells, a mutation is DNA. Mutation is only one of the mechanisms for changing the genome. So we're probably not going to get to the second one. So one mechanism for changing the genome is mutation. The second mechanism is horizontal gene transfer. Genes can be transferred from cell to cell. We touched on that very briefly when talking about crossing over. When chromosomes line up in homologous pairs, they do exchange pieces. So that can happen too. But prokaryotic cells have some very interesting mechanisms for genetic exchange. So tonight we're talking about mutation. And again, mutation is a characteristic of life. It's one of those things where you could just say, mutation happens. Mutation is going to happen. It's just a characteristic of life. What is it? It's a change in the nucleotide sequence of DNA, if we're talking cells. It can be either DNA or RNA, if we're talking viruses. Viruses typically have either DNA or RNA. For a long time, textbooks said they never have both. Turns out that's not true. There's always some new dude that'll show up. It's like, oh, okay, fine. Sometimes they have both. Usually not. Cells have both. Viruses usually don't. Now, some references will define mutation as a heritable change. That is something that can be passed from one generation to the next. I'm going to say that is not going to be our definition. Our definition for this class is a mutation is any change in the nucleotide sequence of DNA, if you're talking cells, DNA or RNA, if you're talking viruses, and most mutations are deleterious, which means they're going to cause damage. If you're talking prokaryotic cells, if you cause enough damage, that cell's going to die. And most of the time, that's what happens. Cells die. Those aren't going to be heritable. That cell can't pass that information along because it's dead. So, never mind that definition. Now, we're going to be looking at some factors that can cause mutations, some chemical factors, some physical factors. Again, this is a section where we're just touching the surface because there's so many things that can cause mutations. We don't have time to look at them all. Most of the time, you don't know what the mutation is coming from anyway. It's kind of like if you have an allergic reaction, and you go see an allergist, and they'll tell you, we don't really know what caused that reaction. You may never know. So mutations happen, but we don't necessarily know what was the specific factor that caused the mutation. So then we say that those mutations are spontaneous. The spontaneous mutation rate is supposedly 1 per 100,000 copies of DNA. That means DNA polymerase can copy DNA molecules, I said 100,000, it's 100 million, 1 per 100 million. 100 million times and make one error. That seems like not much in the way of rapid change, but then think about bacteria for a moment. If it's 1 per 100 million copies, how long does it take bacteria to get to that number? When we did our population counts, we said that bacteria go through a lag phase, they go into an exponential growth phase, they go into a stationary phase. And what was the M concentration? Maximum concentration of bacteria in a batch culture? 10 to the 9th. 10 to the 9th. So that's a billion, 10 to the 9th. So they can get to 10 to the 8th in 24 hours. They reach stationary phase in 24 hours. If you're talking about bacteria that grow rapidly, like E. coli, Staphylococcus, Salmonella, that means bacteria are potentially experiencing one mutation a day. Now, it would be almost impossible to work with E. coli and know that it was still E. coli if most of those mutations weren't deleterious and just caused the cells to die. But they do. Most of them, they're gone. Mutations happen. Spontaneous mutation rate for bacteria, about 1 every 24 hours. Crazy. Of course, mutation rate can be significantly increased, again, either by chemical factors or physical factors, and those things are referred to as mutagenic agents or mutagens. Again, there's way more than we can look at. This little diagram just has a couple of things on there. You're looking at it and going, what are those things in the middle? They look like mealworms. And for a long time, I was looking at that diagram and thinking, what the heck? Cigarettes, okay, I can see that. Benzyl peroxide, okay, it's got a label on it. But what's that thing in the middle? Anybody know? Those are hot dogs. Those are hot dogs because they have sodium nitrite in them, or sodium nitrate in them. Apparently that's a carcinogen also. So mutagenic agents, because they trigger mutations, they can often lead to tumor formation, and so they are often considered carcinogens also. Yeah, I looked at that for a long time and thought, what the heck? Well, anyway, so mutations can be carcinogens, cancer-causing agents. Mutagens, not mutations, sorry. Okay, so we're going to look at some types of mutations, and again, I'm going to restrict this. There's a lot of mutations that happen in eukaryotic organisms we're not going to deal with. So we're looking at mutations as they impact bacteria primarily. If you were dealing with multicellular organisms, mutations can be either germline mutations or they're somatic mutations. Which ones could be heritable? If you experience a mutation in your finger, can you pass that to your offspring? The only mutations that are transferred to the next generation are germline mutations. Fortunately, with prokaryotic cells, you don't have somatic tissue versus germline tissue, you've just got prokaryotic cells. So any mutation is potentially heritable if the cell doesn't die. Another way to categorize mutations is point and non-point. So we're not going to deal with germline and somatic mutations again because we're not dealing with eukaryotic cells. We're dealing with bacteria, archaea. We're talking prokaryotes. We will look at the categories point versus non-point. So a point mutation is a change in a single nucleotide, one base, somewhere on the genome, a change in one base. And again, if the DNA polymerase can copy the DNA 100 million times and just make one error, if it's a point mutation, it's just one base change. If it's a non-point mutation, non-point mutations involve segments of DNA that are considerably larger than that, often multiple genes in length. And we'll talk about some of the ways that non-point mutations can occur, but first we're going to look at point mutations. So point mutations, single base change, that is one nucleotide, one letter, adenine, thymine, cytosine, guanine. Now, if you can change one base, what are the ways you can change it? You can add a base. So if you add one, this can be called an addition. And I don't know why, but more recently, the terminology is starting to change. So these used to always be called addition type point mutations. Now they're called insertion type point mutations. It has to do with people wanting to call these indel mutations, indel insertion deletion. They didn't like addel. Anyway, so additions, you just add one base. This is more commonly being called now an insertion. And the way these diagrams are working down here, they're showing you the original sequence up here, and then they're showing you the inserted base here. So this is being inserted. It's going to cause a frame shift. We'll talk about that in a little bit and what that means. This is the kind of thing I'm talking about. So on the quiz, how many of you have done the homework, the DNA, RNA? Okay, so if you've gotten through that section where you had to transcribe the DNA, and then you had to translate. Transcription is making an RNA molecule using the DNA nucleotide sequence. Translation is then using the RNA nucleotide sequence in sets of 3 bases to encode a polypeptide sequence. I'm looking at, for the quiz on Tuesday, giving you guys the genetic code so we can talk about what happens when you add a base or delete a base or do whatever. Okay, so you need to be paying attention to this and be very familiar with what are we talking about when I say transcription and translation. Okay, if you can add a base, what else can you do? You can take one out. So an addition or an insertion is putting an extra one in. A deletion is taking one out. If you take one out, the little arrow over there to the side again says frame shift. Yeah, you're going to get a frame shift, and we'll talk about what that means. The other thing that you can do is substitute. That is, just change the base. Put in a different one. And a lot of mutagenic agents tend to cause that type of mutation where they will cause a single base change, but the base is just modified. So then we have to look at what is that going to do? And that's where you can play about with doing the transcription and then the translation and see if it makes any difference. So of these mutation types, substitution type point mutations are the least likely to be damaging. Part of the reason for this is because of all the redundancy in the genetic code. So there's a lot of places where you can change one base in a codon and it's not going to make any difference whatsoever. So the other thing about typically substitution point mutations are supposed to be purine for purine, pyrimidine for pyrimidine, but it's definitely not always the case. You can get a pyrimidine for a purine and vice versa. So in this example, here we see a DNA nucleotide sequence. This will encode an RNA nucleotide sequence, which in this case, the RNA is designated as messenger RNA. Everybody knows how to do that, right? I hope so. So recognize again that T codes for A would code for T, except RNA has no thymine. RNA has uracil instead of thymine. So you need to be able to do that. That's the transcription process, getting from a DNA nucleotide sequence to an RNA nucleotide sequence. So you can look at the one and see what the other one's going to be. And then to get your amino acid sequence, you have to read that genetic code in sets of 3. AUG is always the start codon. It encodes the amino acid methionine. We talked about the process of translation where the small ribosomal subunit has to bind to the messenger RNA, and then the transfer RNA carrying methionine actually has to come down and attach before the second ribosomal subunit can bind. Because of that, a substitution can be very significant. We'll see in just a moment. So in this case, again, you would read these in sets of 3. You'd have to have the genetic code, because I'm not asking you to memorize that. I'm also not asking you to memorize the letter sequences for amino acid sequence. So here we're just calling them the amino acids. This is methionine. Some of these are shortened, so methionine, glycine, glutamine. This is phenylalanine. A lot of times you'll see 3-letter combinations that represent amino acids. Well, anyway, so the last codon in this case, UGA. Remember I said that this is you go away. So you go away, you are gone. These are all terminator codons. What's the other one? You go away, you are gone, you are away. Those are the terminator codons. So this doesn't encode an amino acid, that's a stop codon. That's a very short section, but anyway. So here we're going to do a mutation. So here's our DNA. We've changed the RNA. We let this substitution happen. Actually, in this particular instance, this mutation happened in the last base in a codon. If you change the last base, very often, it's not going to do anything. A lot of the codons, if you look at the genetic code, especially if you look at the one that is the circle, if the first two bases of codon stay the same, you can change the last one to any base. And this would be the case here. This could be a G, a C, an A, or a T, or an A or a U, excuse me. It wouldn't make any difference. It's not going to change the amino acid sequence. So if a single substitution type point mutation, all you do is change one base, but it does not change the amino acid sequence, this is called a silent mutation. So it has no particular effect. And this is why, because of the redundancy of the genetic code, this is why substitution point mutations may not have, they can be not serious at all. They have basically no effect. Sometimes. So again, you can see a lot of versions of why this would be, but it has to do with the genetic code redundancy. A lot of codons, if you just change the last base, it doesn't do anything. But now, if we substitute a C for the A in that third position, so we can change one base. Oops. We've changed what was, it was glutamine. Now we've changed it to histidine. To put another one in there. If you change one base and it does change the amino acid, this is called a missense mutation. So you've changed the meaning. Now if you think about this, a polypeptide is many, many amino acids in length. Does changing one amino acid make any difference? Sometimes, again, the answer is no. You can change one amino acid and the protein will still function. On the other hand, sometimes if you change just one amino acid in that polypeptide, you can get a significant change. So apparently the difference between normal hemoglobin and sickle cell hemoglobin is due to a single point mutation. Changing one base, changed one amino acid that made normal hemoglobin not normal, became sickle cell hemoglobin. Is this damaging? Yes, because it causes sickle cell anemia. On the other hand, people who have sickle cell anemia are more resistant to malaria. So it's got multiple functions or multiple effects. Anyway, changing one base can be silent. Changing one base can give you a missense. If we substitute a T for C in the third position of the third codon, apparently there was one there. What the heck? What's labeled there doesn't have any T's or C's. Oh. Yeah, that doesn't make any sense. I think there was a T. It was a DNA nucleotide sequence. There was a T up top. So we took the T out and replaced it with a C. Anyway, what happened is we inserted what was tryptophan. We changed that, and now it becomes a stop codon. So if you insert a stop codon someplace in the middle of a sequence that was supposed to be a polypeptide, you're going to cut that or truncate that polypeptide, and probably it's not going to be functional. If you stick in a stop codon, because the stop codons themselves were for a long time referred to as nonsense codons, if you change a single base and it changes a codon from a functional codon to a stop codon, this is called a nonsense mutation. And again, that's going to be more damaging. So nonsense mutations are going to stop the function of that protein. So if you only make half a protein, it's probably not going to be able to function. Consequently, they're very likely to be damaging to the cell. And then the other thing is if you change a single base at an intron-exon border, we just found out recently, my husband, in fact, the reason that I wasn't here on Tuesday is because my husband was at Stanford getting a CT scan because he had non-small cell lung cancer. Many years ago, he was diagnosed with non-small cell lung cancer, and it was metastatic lung cancer. And as the doctor was explaining to us, you're an anomaly because most people with that situation aren't alive anymore. The other thing they found out is that this lung cancer was caused by a single point mutation at an intron-exon border. So they know exactly where that mutation occurred. Consequently, if the cancer comes back, they said they can make a drug therapy for it. But it's not there. So our trip to Stanford was to find out what was happening, and what's happening is nothing. The scan is clean. So, yeah. The worst thing that happened was the traffic. But anyway, so that I stuck that in there because I thought, wow, a single mutation at an intron-exon border, what does that do? Well, you think about it, then you're messing with post-transcriptional modification. You don't modify the RNA, and you end up leaving something in that was supposed to be taken out. But what would happen if you did a substitution that was supposed to be a start codon? Then you're really in trouble. Again, you have to have that AUG up front in order for the first transfer RNA to come in holding methionine or FMAT. If that doesn't happen, the two ribosomal subunits don't come together, and you can't do translation at all. So a single point mutation that would affect a start codon, again, it's going to be lethal if the cell, if you're talking about a polypeptide, the cell has to have. And again, most genes encode proteins that are constitutive. The cell has to have them. So it's very likely that that kind of a mutation would be lethal. So all of that had to do with substitution. A lot of, you have no effect, or you can have a missense, change one amino acid, which may or may not matter. You can have a nonsense, which is going to be damaging, or you can mess with a stop codon or start codon, that's going to be way damaging. But now we're going to look at additions and deletions, because additions and deletions always have the same kind of impact. If you add one extra base, or insert one extra base, or if you take out one base, you're going to get a shift in the codon reading frame. Remember that translation happens by reading the bases in sets of 3, and the cell does it automatically. The messenger RNA is coming through the ribosome. When that codon is coming in to the A site, you're going to get a specific amino acid that's going to bind at that location as directed by the sequence in the messenger RNA. So if you take out one base in the DNA or add one extra base in the DNA, it's going to shift the codon reading frame, either to the right or the left, depending on what you're looking at. So this is called a frame shift mutation, where a substitution may or may not be damaging. A frame shift is going to be damaging. It's going to change all the amino acids beyond that mutation point, or probably all of them. So this is just showing you some specific. You have the sequence that's up above. You can see the position of the codons, and then we add a single base at the bottom. The codon reading frame is shifted one base. The same thing is going to happen if you delete one. It's just you're going to shift the codon reading frame in the opposite direction. The first time it was shifted to the right, the second time it's shifted to the left. I'm not going to ask you, does this shift to the right or to the left? It doesn't matter. A frame shift means you're going to shift the codon reading frame. You're going to get a brand new amino acid sequence. So, could it still be a functional protein? The likelihood of this is not great if you suddenly just change the amino acid sequence. On the other hand, it's not impossible that you couldn't have a protein with a brand new function within the cell. Mutation happens, and when mutation happens and you get a shift in the reading frame and suddenly you get a brand new amino acid sequence, maybe what's created is a protein with a novel function, a function that didn't exist before. As I said, mutation is a characteristic of life. If you get a novel protein that has a unique function, then whether or not this is damaging to the cell depends on what that function is and what's going on in the environment around that cell. So let's look at a situation where we have a bunch of bacteria. In this case, they're all blue and there's one pink guy. That pink dude is a mutant. So it has experienced some kind of a mutation. Let's assume that that was a point mutation that allowed that pink cell to produce a brand new protein. So it was a frame shift. It was either an insertion or a deletion. We get a shift in the codon reading frame. We have a brand new protein with a brand new function. This particular protein is going to be an enzyme, and the enzyme is going to be called beta-lactamase. Anybody know what a beta-lactamase enzyme does? This is a pretty common functional enzyme associated with bacteria that are resistant to beta-lactam antibiotics. The beta-lactam antibiotics are a whole bunch of antibiotics that have in their structure what's called a beta-lactam ring. All penicillins and all cephalosporins have beta-lactam rings. If bacteria produce beta-lactamase, they can break that ring, and consequently they can take these drugs apart. So let's say that this particular group of bacteria, and that one little mutant there, find themselves within a patient that is taking beta-lactam antibiotics. What's going to happen to the population? The pink guy is the only one that can produce beta-lactamase. The rest of those guys are going to die. They're going to get wiped out by the drug. And we've got one little bacteria left, and what's that one going to do? It's going to reproduce. And now we have a brand new population, and they're all resistant to beta-lactam antibiotics. So the entire population has become resistant. This is happening over and over and over again with different types of drugs, particularly if a person has some type of, well, what happens is if they're given only one antibiotic at a time, the bacteria will, if they keep mutating, eventually they will develop resistance. So it's just because mutation happens. That happens. This process, by the way, is called evolution. Change in populations over time. Because populations mutate and there's selective pressure, in this case the selective pressure was the drug being in the environment. But there are lots of drugs in the environment because they're being given to patients, and patients use bathroom facilities. And they're releasing quantities of these drugs into the environment. They're getting into the water supply. Apparently, they're all over the planet. Lots of antimicrobial drugs, and as a consequence, lots of bacteria are exposed to these. When penicillin was first developed, so remember Alexander Fleming and his work with penicillin, he was the one that first identified this drug that would control staphylococcus. It was highly effective during World War II. Now, penicillin G, most staphylococcus are completely resistant to it. The good news is the one we work with in this lab isn't. It's still susceptible, which is good. But that's not characteristic of staphylococcus out in the real world. So again, this is natural selection, where you have organisms, they experience mutation. If there's a selective pressure in the environment, which in this case is some antimicrobial drug, the cells that are susceptible to the drug die, and the ones that are left are the ones that survive, and they then change the population because they're the survivors. And this is happening over and over and over again. I stuck this in here because at not this last micro conference, but one I was attending earlier, they were talking about this problem and how significant it is in the healthcare setting and what to do about it. And so now what they've done is they set up working groups within hospitals that are antimicrobial stewardship teams. And their job is to try to determine, are antimicrobial drugs being used responsibly? By patient, should this patient be given this drug or not? Is this really going to be effective, or is this just leading to more resistance down the line? They're trying to curtail the use of antimicrobial drugs. Unfortunately, I really think the biggest problem in this country isn't the clinical use of antimicrobial drugs, it's the agricultural use of antimicrobial drugs. Because we blanket drug lots and lots of animals that are being used as food animals. They're given antibiotics because they grow faster. Well, that's all fine and good, but if they're the same antibiotics that you have to use in a clinical setting, and now the bacteria are all resistant to them, that's not a good thing. And that's kind of where we are. Apparently in Europe, they have determined that this is a severe enough problem that they've stopped using antimicrobial drugs in agriculture. They said, stop it. You can't keep doing this. In the United States, we still do it. And supposedly we're looking into it. We're looking to see if it has an effect. Sure it does. But somebody might not be able to make money fast enough. So drug resistance is a serious, serious problem. Okay, now we can look at some non-point mutations, and then we'll come back and look at some mutagenic agents. Again, non-point mutations involve regions of DNA that are considerably longer, so they're often lots of nucleotides, sometimes more than one gene in length. One way that this type of mutation can occur is through what's called a translocation or transposition. I should warn you guys up front, watch out for the trans words. Okay, so we had transcription and translation. Transcription is RNA synthesis. Translation is protein synthesis. Now we're going to have translocation and transposition. This just means you're moving a segment of DNA from one location to another, and this can be either within a chromosome or between chromosomes and plasmids. It turns out that these translocations or transpositions sometimes involve segments of DNA called transposons. Transposon, I think, is short for transposable element, but transposon is the most commonly used name for this. So a transposon is a segment of DNA that can initiate its own translocation or transposition. These things move. That's why I said earlier, a gene has a specific locus, but they don't necessarily stay put. They can move to some extent. So this is a diagrammatic representation of a transposon in bacteria. It's telling you that the blue region is a series of structural genes, and at either end, there's what are called inverted repeats. These are particular segments where an enzyme can cut the DNA. It's an enzyme that is associated with the transposable element, and consequently the enzyme itself is called transposase. So again, transposons are these segments that can move. Another name for them is jumping genes. Apparently the term jumping genes was coined by a lady by the name of Barbara McClintock, and she was studying corn plants and trying to do Mendelian genetics with corn plants and doing these genetic crosses, and came to the conclusion that genes didn't stay put. They were moving. And she said, some of these genes are jumping, these jumping genes. Well, it turns out that's exactly what they were. These transposable elements or transposons move. Again, at least, what's associated with this, what's cutting, and it cuts in that region of the inverted repeats on the ends of the transposon. These enzymes called transposase enzymes. So they recognize the DNA, they cut it, and then they let the DNA move and go to a new location. Apparently the enzymes themselves are not necessarily encoded by genes that are in the transposon. They can be encoded by genes someplace else. And these things are fairly common. What's interesting is that transposons are often associated with genes for antibiotic resistance, and because these genes can move from location to location, they often, transposition or translocation, as I said, can be from chromosome to a plasmid, and plasmids can be transferred to other cells. And because of this, bacterial resistance is often transferred. One cell gets resistance through mutation, and then they transfer that resistance to other cells through horizontal gene transfer, which we talk about after mutations. This is just a diagrammatic representation showing you how the transposon moves. So you have the transposon, that little red section, and then the inverted repeats on the end is where the transposase enzyme comes. Again, it's going to break phosphodiester bonds. It's going to allow that little segment of DNA to be cut out. If it's going to move to a new location, then it has to go to wherever it's going to go, and again, the cut occurs, and this thing gets incorporated. Sometimes they get incorporated, sometimes they don't. So you can imagine that transposons could be responsible for rather large losses or deletions of segments of DNA. And if they do get into a new place, that's a rather large addition. Anyway, this is just talking about a sequence called ALU that is in the human genome. So in Biology 1, I think they do ALU, and they look for ALU insertions. There's about 300, this thing's about 300 base pairs long, and there's lots and lots of these within the human genome. So these things have been inserted in multiple locations. Exactly what their function is, I don't know, or do they really have a function? They're just something that people recognize. Transposons are not restricted to prokaryotic cells. Genes do move about. The other place translocation occurs is during meiosis, when chromosomes line up in homologous pairs and they cross. The crossing over that happens. Typically you'll see this diagrammed as a rather simple type of crossing over. So here we see purple chromosome, orange chromosome down here at the bottom, and you're seeing just this bottom section crossing, so that the purple chromosome picks up an orange segment and the orange chromosome picks up a purple segment. This is way simplified because crossing over is much more common. There's a lot more DNA exchange during crossing over. But again, that's considered a form of translocation or transposition. So it can be caused by transposons that move using transposase enzymes, but not always. Well, if you can have a piece move from one place to another, as I said, these transposons, when a transposon comes out of a chromosome, there's going to be a space. So that's a deletion. Removal of a segment of DNA, one or more genes in length. Which that diagram is way too small. It's just showing you a little bitty deletion that's only three letters long. So transposons can cause deletions. There's some other things that can cause deletions, which we'll get to later. And then another type of non-point mutation is called an inversion, where a piece of DNA is just turned upside down within the chromosome. if the DNA is looped, if it makes a loop and then it's broken and reattached, you can have a section of DNA that gets turned upside down. These breaks don't necessarily occur right at the beginning or right at the end of a gene, so they can happen in the middle of a gene, in which case you may get the front end of one gene connected to the back end of a different gene. The likelihood of producing a functional protein, you might think, well, that's a highly unlikely thing to happen. But then think about words, particularly all these words that we have that start with trans and end with TION. You've got transcription, translation, transposon. Well, that's not, it's got a different ending, but you have a lot of words that begin with trans. So if you think about segments of DNA having the same kind of thing, apparently genes have specific regions. If you take that region and connect it up with some other ending sequence, you might still have a functional gene. There's a lot of variation that goes on. So these mutations that involve large sections are most likely to be lethal, but they're not always going to be lethal. Again, because of this recombination business. Which reminds me, when I was talking about post-transcriptional modification where introns are taken out and exons are connected together, apparently new information suggests that the exons that are connected together aren't even from the same gene. So in eukaryotic cells, pieces of genes can get recombined during that process also. And then we didn't talk about post-translational modification, but that happens too. There's a lot of weird stuff that's going on with molecular genetics. Okay, so now let's take a look at some mutagenic agents and see how far we'll get with this. Again, this is a huge topic because there's so many things that can be mutagens. So we'll just look at some specific examples, a little bit about mutagens. And again, mutagenic agents are just factors that are going to increase the mutation rate. If the spontaneous mutation rate is 1 per 100 million copies of the DNA, a mutagen is going to make that more common. Mutations are going to be more common. They cause mutations. How do we know if something is a mutagen? Well, there's a test that can be run. It's called the Ames test. And basically, this employs a method where bacteria, specifically Salmonella, are subjected to chemicals. Typically, they're tested one at a time. And then you look at the Salmonella after the exposure to the chemical to see if they have a significant number of changes in their metabolism. So here you see the Salmonella, you see the possible mutagen being added to it, and then you see what they have here as a control. This is considered the natural mutation rate. This is a much higher mutation rate up here, so whatever that chemical was, it's a mutagen. If you've ever read the book, The Emperor of All Maladies, it's a book about cancer, and it talks about carcinogens, these chemicals that might be mutagenic agents. And it says this kind of a test is all well and good, but it has a significant drawback, and that is that these chemicals are being tested one at a time. And that is not how we're being exposed to them. We're being exposed to them in complexes because there's so many chemicals that we're being exposed to now that we're exposed to multiple ones all at the same time. So we really don't know how they affect us when they're working together. Nevertheless, the Ames test is one of the ways that you can test a chemical to see if it's a mutagen or not. Now, mutagens are not always chemicals. Mutagens can be physical factors. We'll talk about some chemicals. We might get to some physical factors. We'll see. So chemical mutagens, again, these are chemicals that are known to increase the mutation rate. And you can test them using the Ames test. There are a group of chemicals called base analogs. This is a kind of a large group of mutagens. There's a lot of specific chemicals that fall into this category. A base analog is a chemical that is structurally similar to a naturally occurring base. So the bases are adenine, thymine, cytosine bonds. A base analog has a structure very similar to one of those, and consequently can be incorporated into DNA in the place of one of those. So here we see two molecules. One is thymine, and the other is a compound called 5-bromouracil. Thymine is 5-methyluracil. So you see that CH3 up in the corner? Otherwise, if you took that CH3 off, that thymine would be uracil. So 5-methyluracil is thymine. 5-bromouracil is a base analog. It can be incorporated into the DNA molecule in place of that thymine. Now, when the DNA replicates, it depends what form that mutagen is in. It can either code for the proper complementary base, or it can code for the wrong one. So normally, thymine would encode adenine, and you see thymine, adenine, or in this case, the keto form of 5-bromouracil forming a normal bonding that would have been thymine to adenine. And then you see it down here doing a slightly different version because it's in the enol form where it can form 3 hydrogen bonds with guanine. What kind of a mutation is that? The normal base that would pair with thymine would be adenine. If 5-bromouracil encodes guanine instead of adenine, what is that? Just in general, that's a substitution. So that's going to give you a substitution type, point mutation. Furthermore, it's still purine for purine, but again, it's not always like that. So uracil is a pyrimidine, thymine is a pyrimidine, adenine and guanine are purines. So in this case, adenine is replaced by guanine, and you get a substitution type point mutation. And as we saw, substitution may or may not be damaging. Depends on where it happens. But they actually use 5-bromouracil as a anti-cancer chemical. It can be used topically, and it's supposed to cause rapidly growing cells to die. It causes the surface of your skin to become seriously damaged if you have precancerous tissue in there. I don't know what it does to your bacteria, but it can't be good. So that's a base analog. There's other types of analogs. So this is a nucleoside analog called azithromycin or zidovudine. This is an anti-AIDS drug, and it actually has a specific function in that it inhibits the enzyme reverse transcriptase, which is what allows retroviruses to reverse transcribe RNA into DNA. Another type of chemical mutagen is an alkylating agent. And an alkylating agent is a chemical that's going to add methyl or ethyl groups to bases, and then that can change how those bases hydrogen bond. Now, don't worry too much about specific names of alkylating agents. Just recognize that an alkylating agent changes a base by adding a methyl group or an ethyl group. When it does, it changes the way that base hydrogen bonds. So guanine would normally encode adenine, but this methylated guanine is going to encode thymine instead. In that case, you're getting a substitution that is pyrimidine for purine. So again, it's a substitution type point mutation. The other thing alkylating agents can do is they can cause bases to bind to each other. This little green line in here, it's kind of hard to see, but what it represents is covalent bonding between those two bases. That's not hydrogen bonding, that's covalent bonding. So those bases are stuck to one another. When that DNA replicates, instead of having two bases that code for two complementary bases, that thing that's stuck together is going to encode one. What kind of a mutation is that going to cause? Where two bases are now going to encode one complement instead of two. A deletion. So alkylating agents can cause deletion type point mutations. We'll get to it. Deletion, yeah. This is the other type of mutation that these guys can do, where they can cause bases to covalently bond across the DNA strand. If that happens, that DNA molecule can't open. The enzymes the helicase that would cause the two DNA strands to come apart aren't going to work. The DNA strands don't come apart. Consequently, the DNA can't replicate and it can't be transcribed. So it's pretty dysfunctional. Alkylating agents with that kind of activity are definitely going to be lethal. And they can cause destruction, etc., etc. And we're out of time, so never mind. I think the next thing is physical factors. We'll talk about physical factors next time. Okay. So we do have a quiz tonight. Hopefully everybody was ready for that. If not, you've got 10 minutes. And then we have a fun lab to do. I need to make sure we've got all our stuff.